Well, good afternoon, March for Lifers. Thank you so much for joining us today. We have a very special guest for you. Dr. David Prentice is the Vice President and Research Director for the Charlotte Lozier Institute. He's also the Adjunct Professor of Molecular Genetics at the John Paul II Institute, the Catholic University of America, and an advisory board member for the Midwest Stem Cell Therapy Center. Dr. Prentice is an internationally recognized expert on stem cell research and bioethics, and has provided scientific testimony before the U.S. Congress and numerous state legislators, the U.S. National Academy of Sciences, and the President's Council on Bioethics. So thank you for joining us, Dr. Prentice.
Great to be with you, Anne Claire. Thank you.
It's a mouthful to rattle off all of those accomplishments. No, it really is wonderful to have you joining us. I feel like this is such a timely topic that we'll be diving into today, and it really is an honor to speak with you about it.
Glad to help educate, because as you mentioned, this is a topic that not a lot of people are familiar with, or they've heard a little bit, but we will do not a deep geek dive, but we will talk some science and biology and try and demystify a lot of this, but also mention the ethical connections here for vaccines.
Mm-hmm. Well, then diving right into it. In the midst of the coronavirus pandemic, vaccine production is definitely a timely topic. But can you explain why this is such a pressing topic for the pro-life community?
The short answer here is because some vaccines, both some vaccines that we have currently, as well as some of the vaccines that are being developed for COVID-19, make use of abortion-derived cell lines. So fetal cells, now they've been in culture for years, but they were originally derived from an abortion. And so there's an ethically controversial aspect to some of these vaccines. And we'll spend some time during our chat today going through how vaccines are made and what kind of cells, if any, are used. But the key point here is that connection to abortion. And as I mentioned, there are some current vaccines that have been used over the years, where they grow virus in these abortion-derived cells. Then they harvest the virus and kill it or inactivate it in some way, and that's your vaccine. Mm-hmm. Well, there's an ethical question there because of that line back to an abortion. And it may have been decades ago, but there is still an ethical line that can be drawn. And for some people, that brings up a crisis of conscience. Can they avail themselves of that particular vaccine?
No. Well, thank you for that explanation. So when reading through a lot of the arguments and articles on vaccine production, you see a lot of bioethicists who have justified using fetal cells because they're from abortions that occurred decades ago. I believe the main lines were almost 60 years ago. Or others argue that vaccine production is ultimately for the greater good of our society, which outweighs any other ethical qualms. What are your responses to these debate points?
And this has been debated, you could say, for years by bioethicists, looking at that ethical link, that line, as we can say that's drawn back, and it could be decades. Some of these cell lines that are used even today were derived from an abortion that occurred in 1962. There's some from the 70s and a couple from the 80s. But yeah, so it's been a lot of time. And the argument has been from some that because it has been a long time, and as you note, for some because of the potential greater good, that it's okay. It's interesting that both Catholic and Protestant scholars have looked at this question, and usually what people look at is just that first response, that it's okay. but they haven't really looked deeply enough or looked at the second part of the answer. So in the Vatican, the Pontifical Academy for Life in 2005 wrote about this question. It was also written by the Congregation for the Doctrine of the Faith in 2008 in Dignitas Personae. And what they say basically is if there is a grave reason, in other words, you're trying to protect a child from some life-threatening disease, that you can, in good conscience, use that vaccine. But note what I said. It wasn't just, we're just dismissive and because it's been a lot of years. It was if there is a grave reason that you can use that vaccine. There's also a couple of other parts to what was said, especially in Dignitas Personae, that if there's a grave reason and there is no licit alternative. So maybe you're faced with a choice and there are two different vaccines produced. One has used this ethically questionable route of production, and another has used an ethically uncontroversial route. We ought to probably be opting for the alternative where there is no controversy, unless there's some grave reason and you can't access the alternative. And then there's another part, and that is even if you are facing this and because of the grave reason and no alternative, you access that vaccine, you still should be advocating to your doctor and to the pharmaceutical industry that they produce a licitly developed vaccine. And so, you shouldn't just be brushing this off. People need to be considering this, and it has to do primarily, we're talking about vaccines now, but in a number of pharmaceutical productions, whether there will be some sort of ethically questionable production method. People need to be looking into this.
Well, in that same vein, you recently authored an article, A Visual Aid to Viral Infection and Vaccine Production, which was a phenomenal read. It brought me back to my days in biology courses. And one of the elements that really struck me was that for every method of vaccine production, human fetal cells don't seem to be required. For example, you mentioned the use of placenta or even viral monkey cells. If this is the case, why are then we seeing research labs using human fetal cells, considering so many individuals have moral or ethical qualms against this practice?
Great question. And there are several parts to the answer, but one part is simply this is the way we've always done it. And so we've always used those cells, speaking as the pharmaceutical industry or even a scientist in a lab developing a potential new vaccine. They've always done it this particular way, and they're sort of set in your ways. I mean, scientists are people too, and we can have certain human failings along those lines. There might be a question of economy. Maybe they haven't spent the money to develop a new procedure and so on. And we'll go through some of those in a minute. Obviously there are some people who have no ethical qualms, and so it may not even occur to them that there could be an ethical controversy about some of the production methods. And in fact, even people in good faith who would have a qualm may not know about the derivation of a particular cell line. So there's very much an educational hurdle that we need to get over, not just for the public in general, but even for the physicians and the scientists.
And do you think that pharmaceutical companies or the research labs that are investing their time and resources into vaccine production have a responsibility to our society to protect people from using vaccines that might go against their own ethical standards?
It's hard to say where you might consider that they have a responsibility. I think it's a responsibility on people of conscience to make them aware of that particular potential problem, because and as well as policymakers, not just the vaccine producers, but the funders and so on. Simply because if you're looking for the greatest application to protect the public health, then you want the whole public to be able to access a vaccine in good conscience.
Mm-hmm.
And so it behooves them to consider these ethical questions and not just safety and efficacy and the economics.
Oh. So in June 2019, the US Department of Health and Human Services announced that it would no longer provide intramural funding for government research that requires new acquisition of tissues harvested from victims of ongoing elective abortions. However, when reviewing many of the COVID-19 vaccine programs, you see the use of the HEK 293 and the PER C6 cells in programs that are receiving the majority of HHS funding. Looking into this, you're seeing millions, and in some cases billions, of US tax dollars going in to research using these fetal cells. Why is this currently being allowed?
Mm-hmm. And here we kind of need to step back and look at what that HHS decision had to do with. And frankly, it didn't have anything to do with these abortion-derived cell lines or historic cell lines at all. Uh, what that decision was about was what some have termed fresh fetal tissue, but
Mm-hmm.
fetal tissue being derived from ongoing abortion. We're talking about tissue. We're talking about brains and hearts and livers and other aborted baby body parts. Uh, if if your listeners have seen any of David Delyden's videos, that's what this decision was about, and that's what the debate was about at that point in time. So, and what the HHS decision said was that federal taxpayer dollars that were going towards that funding, there were going to be some restrictions put on. Number one, the federal government would no longer use taxpayer dollars for fetal tissue like that in what's termed intramural research. In other words, research that's going on within the confines of government laboratories. Extramural research would be somebody at a university lab, let's say, who gets a grant to do a certain type of research. And there are federal taxpayer dollars going towards that type of research with fetal tissue. Another part of that decision was that any new grants or any renewals, if somebody wanted to come back and renew the research they were doing, would have to go through an ethics review board. If I write a grant to do some kind of research in my lab, it goes through a scientific review, and then hopefully I get funded to do the kind of research that I propose to do. Well, this would go through an additional review having to do with the ethics of the research. And the Health and Human Services Department will be impaneling this particular ethics review board to look at this in the future. The other thing that they said in that decision was they would start to put money towards non-fetal alternatives in terms of research, whether that was adult stem cells or induced pluripotent stem cells, other types of cells, licitly derived, tissue derived from surgeries, on and on and on. Mm-hmm. Again, all this was about the fetal tissue aspect. Separate issue over here, even though, yes, there's an ethical line you can still draw on, were these historic cell lines, these abortion-derived cell lines. And I might point out, we kind of passed over the fact that there are some vaccines currently, not just in production, but that have been used for years, where they still use some of those old cell lines.
Mhm.
A small handful, and they're moving away from using those, but there are still some of those that people face, and a couple in which there's no alternative.
Mhm.
Uh, and then we've got, and you mentioned the cell lines. That's great. You're getting into the real geek stuff here when you start naming cell lines. HEK 293, and the HEK stands for human embryonic kidney. It's again derived from an abortion, maybe years ago, but
Mhm.
still, you've got that ethical line to trace back. And then PERC6 is another abortion-derived cell lines. Those two are the ones that are the focus now because of some newer ways that people are trying to make vaccines. So, if you don't mind, if we kind of just go through real briefly, and for the listeners, and Clara said she'll put a link up to our primer on virus infection and ways to make a vaccine. But let me just give you sort of a thumbnail verbal sketch now. When a virus infects a cell in my body, when I have that type of viral infection, viruses, in a sense, aren't even alive by themselves. They have to get inside and hijack the machinery of a cell to be able to make more viruses and cause an infection. So, viruses usually have a little bit of genetic material. It might be RNA or DNA inside some sort of coat. And it might be a lipid, kind of like an oily layer. It might just have some proteins. The virus gets inside the cell, delivers that nuclear material, if you will, as genetic material, but it literally does hijack a cell so that it takes over the production scheme to make more copies of its genetic material and to make the proteins that are viral proteins that it needs to get out and then infect another cell.
Mhm.
So, the way our immune system reacts to a virus or any sort of invader, but especially a virus, is it's looking at usually that outer surface of the virus, the proteins that are sticking out, and trying to see, it recognizes that as foreign. And your listeners have probably all seen those great little diagrams of the COVID-19 virus, and it's got all those little knobbies sticking out. Those are called spike protein. The virus uses that to infect the cell, but it's also then the face that virus is showing to the outside world, including to our immune system. And so most of the targets for the vaccines that are under uh, development right now, and I checked a couple of days ago, we were up to 169 vaccines under development. Now, most of those are probably going to fall by the wayside, but they all fall into about five major categories in terms of: how do I make a vaccine? How do I stimulate my immune system so that it's armed and ready if a real virus ever comes into my body to get rid of it? The old-fashioned way, tried and true, it's a proven technology, is to grow that virus in a cell, which leads us back to those abortion-derived cell lines, as well as, like you mentioned, there are plenty of alternatives. There are placental-derived cells. There's a monkey cell line that's been used for a lot of virus growth and so on. But you grow up a bunch of virus, and then you collect that virus and you weaken or you kill it. But essentially, what you get in that vaccine injection is the whole virus, but killed so it shouldn't actually cause an infection. So you're essentially putting that wanted poster up there and you're showing the whole face of the virus. Your immune system sees that, makes antibodies, and gets other immune cells activated so that it's armed and ready should a real infection occur. But now we've got modern ways, or newer ways, we'll say, to make a vaccine. And another one is to package a bit of viral genetic material, and it might be just that gene for that spike protein, the face that the COVID-19 virus shows. But you're trying to get that into our cells, so our cells will make the spike protein. But there are different ways to do it, and one of the ways to do that is to package it in a so-called harmless virus. One of those is what's called adenovirus. It's a virus that causes some of the common colds that we get. But the problem is then you've got to grow the virus in a cell line, and back to those abortion-derived cell lines, the HEK293 or the PER-C6. Some of these vaccines that are under development and steady, they use those old cell lines to grow this viral vector, this sort of carrier. And so there would be problems. You can use other cells, though. And again, the placental-derived cell lines, there are some newer developed cells that are called iPS cells that can be used to do the same types of things. Then there are even newer technologies, you might say, where you don't make the nucleic acid in a cell or the genetic material, but you make that particular viral protein, and you have a cell in your laboratory or in the facility where they're making vaccines, and it just keeps making lots and lots of the spike protein. And again, so you've got a cell that's involved at some point making viral protein. It could be an abortion-derived cell line where there's a controversy, or it could be something else. In fact, and some of the companies now use insect cells to pump out a bunch of this protein. But now what you get in the vaccine is not whole virus, and not even a sort of a carrier, a postman virus, if you will, but just the protein. Again, our immune system looks at that and it says that's foreign. We'll make antibodies. We'll be ready in case that virus ever shows up. A couple of other newer techniques are to give, instead of the protein itself, give the little piece of RNA that codes for the protein, and then let our own cells make the protein in our body, show it to our immune system, or to give a piece of DNA that would code for the protein. And again, our cells are going to be going through the molecular process using our machinery to make the protein in our bodies and show it to the immune system. So, as you pointed out, in none of these cases do you have to use one of these older cell lines. There are plenty of alternatives, but you've got to kind of look and see, out of all of these 169 potential ones, which ones are using a controversial cell line versus which ones aren't. Or some, as I said, don't even use cells at all.
Well, thank you for that breakdown. So you mentioned before that the HHS stated that it would provide funding to optimize and develop the alternative research methods that don't use fetal stem cell lines. So do you see these types of regulations positively affecting the ethical production of vaccines?
Well, and again, those HHS regulations had to do with the ongoing abortion, fetal tissue, and not really the cell lines. But just being aware that there's an ethical question, and that there are people who are wrestling with this conscience question about whether to make use of a product made using a controversial versus an uncontroversial thing. I think that can't help but open up other alternatives and possibilities.
Mhm.
And the ethical question is really, in a lot of people's minds, the most important question. For some people, it'll be safety. For some people, it'll be whether or not. And I might point out there's kind of a race going on here. We don't know who's going to win, but of all of these 5 different techniques around the world to create a vaccine, and just kind of opening people's eyes to the potential for an ethical question that might come up, I think has to somehow steer these along in a better direction. So, the other thing I didn't really point out, but I mentioned these 5 different techniques.
Mhm.
Some are newer. Uh, some of those have never yet made an approved vaccine. So here's another sort of question about if you're going to be sort of spreading your money around, let's say,
Mhm.
and we're placing bets on all these, you want to place your bets on all of the different techniques as well, so that you have an opportunity to come up with something that will actually produce a usable vaccine. Laying that ethical question on as well about, then, you know, will all the public have access to that vaccine and be able to avail themselves of it?
Mhm. And then how can the general population, or what can the general population do in order to promote ethical production of vaccines? Likewise, how can we stay informed on whether our vaccines, whether they're for COVID-19 or other viruses, how can we be sure that they're being ethically produced?
Yeah, and I think there's several ways. We mentioned before that one of the caveats in terms of the ethical decision that Dignitas Personae mentioned is that you might make this decision, but you still need to be advocating for licit production methods, not just for vaccines, what we're facing now, but for any sort of pharmaceutical product. And you talk to your doctor about that. Um, you really should contact the pharmaceutical companies and let them know. I mean,
Mhm.
there are all these companies' names in the news right now in terms
Mhm.
of the Warp Speed initiative. And I might mention that uh, so far, those are all kind of the newer techniques. There's not that old tried and true method yet that's been put into the mix, but that may come.
Mhm.
And there are some questionable, controversial methods being used, as well as some uncontroversial. So, advocating to the pharmaceutical companies, I would say, also to your elected representatives at the federal
Mhm.
level. Let them know that you're at least aware, and hope that they're aware, that there can be some ethical questions and decisions that need to be made very shortly, maybe, in terms of some of these COVID-19 vaccines, but others along the way. And I think also, you know, within your own peer group, your
Mhm.
friends, your family, talk about it with them as well, because the more voices that can be raised to get ethical, successful types of alternatives put forward, the better the chance that it will happen and happen soon.
Thank you for that. And if our viewers are interested in contacting pharmaceutical companies, contacting their elected representatives, what are some resources that they can be pointed towards, that they can learn more about vaccine production, ethical vaccine production, get lists of pharmaceutical companies that are producing ethically questionable vaccines, or doing ethically questionable research?
Well, we mentioned our virus and vaccine primer at the Charlotte Lozier Institute website, which is lozierinstitute.org, but I know you'll have a link up. We've also done an analysis of some of the the front runners, if you will, in terms of these vaccines in another paper that a couple of scholars did, and tried to raise awareness in terms of the ethical issues there that need to be considered, and go through at least those that are in clinical trials or close to clinical trials at this point to sort of demystify some of that.
Mm-hmm.
So again, and Claire's going to have the links for people to go to learn more about viruses and vaccines.
We will be adding that link right after the close of this interview. And so as we're closing out, how can our viewers, if anyone's interested in following your work, best get in contact with you or read up on any of the work that you're putting out there?
I think, again, back to the Charlotte Lozier Institute website. We put various news things up there, or recent articles and so on, and try and keep up with all of our scholars that are working there. And you can leave a message or leave a question there as well.
Thank you for that. And thank you so much for all your time and research that you've put into this topic. You've said it several times throughout, but this is such an important topic for pro-lifers, and it doesn't usually get that much attention.
Right.
But I'm very happy that it's been brought to people's attention in current times.
Well, thank you. Thank you for helping make people aware of this.
Of course. Well, again, thank you for your time. Thank you for all of our viewers' time today. We encourage all of you to reach out and look up information at the Charlotte Lozier Institute. They're a fantastic resource for really any policy or scientific information in the pro-life movement on the life topics. So again, that's the Charlotte Lozier Institute. Thank you again for your time and have a wonderful day.